Hello, I'm Professor Sims, and in this video I'll discuss cell communication and reproduction. This is the fifth in the series of 10 lessons held as part of my General Biology 1 course. If you're a student currently enrolled in this course, please consult the syllabus and course Moodle site for assignments, quizzes, due dates, and other course information. The learning objectives for this unit include describing cell signaling mechanisms, how cell signals directed, protein expression, cell growth, apoptosis, understand the concepts of genes, genomes, chromosomes, chromosome compaction, all of the stages, phases, and control mechanisms of the cell cycle, uncontrolled cell growth, tumors, cancer, and then finally prokaryotic cell division. Cells communicate with each other via cell signaling. Basically, it's how they talk to each other. It's how they interact. Cells need to communicate, to work together, and respond to their surroundings. For example, immune cells chat to each other in order to recognize and fight off infections. Now, there's two types of cell communication: intercellular signaling, signaling between cells, and intracellular signaling inside of a cell. Cells can communicate through direct contact, chemical signaling, or electrical signaling. Direct contact is like a high five between cells, right? They're touching each other. While chemical signaling involves releasing special molecules that bind to receptors on target cells. Electrical signaling works through the flow of ions across a cell membrane. Each type of communication has its own important roles in keeping living organisms functioning properly. Now, the chemical signals are released as little molecules called ligands. Ligands are kind of like messengers. They bind to a specific protein called receptors on a target cell. Think of it as a key fitting into a lock. Different ligands have different receptors, and they're very specific in regards to which receptor they will bind to. There's 4 categories of chemical signals: paracrine signaling, endocrine signaling, autocrine signaling, and direct signaling across gap junctions. The main difference between these types of signaling is how far the signal has to travel in order to reach its target. It's kind of like a game of telephone, but it's all happening inside your body. And not all cells are affected by the same signals. They each have their own preferences. This is figure 9.1, and it's showing the different types of signaling. When cells are close together and they send signals to each other, we call those signals paracrine signals. So that's these here. These signals move by diffusion through the extracellular matrix, kind of like how smells spread through the air. They cause a quick response that doesn't last very long. And in order to keep the response contained in a specific area, the molecules that carry the paracrine signals are quickly broken down by enzymes or taken away by neighboring cells. By getting rid of the signals, it allows them to spread out again if they're released once more. Let's take an example of paracrine signaling. Figure 9.3 shows the example of paracrine signaling a nerve cell synapses. So nerve cells have a cell body, and then they have dendrites that receive stimuli, and an axon that transmits the signals to other nerve cells or even to muscle cells. The synapses is the junction where the cell transmission occurs. A synaptic signal is a chemical signal. It travels between nerve cells. Signals within the nerve cell propagate through fast-moving electrical impulses. And when these impulses reach the end of the axon, the presynaptic cell releases chemical messengers called neurotransmitters. The neurotransmitters travel across very short distances known as chemical synapses to reach the next nerve cell. The close proximity allows for quick signal transmission, enabling immediate responses such as withdrawing your hand from a hot stove, for example. So after they bind to the receptors on the surface of the postsynaptic cell, the neurotransmitter causes a change in the electrochemical potential of the target cell, launching the next electrical impulse. The released neurotransmitters are quickly degraded or reabsorbed by the presynaptic cell, and this ensures that the recipient nerve cell recovers quickly and remains prepared to respond rapidly to the next synaptic signal. So that was a lot, but it's illustrated beautifully in this figure, and it is just one example of paracrine signaling. Now let's go back and discuss endocrine signaling. This type of signaling involves cells sending messages to distant cells in the body. So the cells responsible for these signals that go far away, right, that's called endocrine cells, and they hang out in glands like the thyroid, the hypothalamus, the pituitary gland. The interesting thing about endocrine signals is that they bring about slower responses, but they have longer lasting effects. The chemicals released in endocrine signaling are called hormones, and they're like little messengers produced in one part of the body that have the power to affect other regions of the body that are quite far away. So that was endocrine signaling. Okay, so the distance is much further, but the effect is much longer lasting than paracrine signaling. Now, autocrine signaling, this is a way that cells communicate with themselves. The signaling cells release molecules that actually bind to their own receptors. And this kind of signaling is crucial during early development to make sure cells become the right kind of tissue with the right function. It also helps to regulate things like pain sensation, inflammatory responses, and even programmed cell death, which we'll talk about some more, such as in the case of an infection or something like that, right? Sometimes the neighboring cells of the same type can also be affected by released molecules of the autocrine signaling system. It's like a group chat where everyone gets the memo. This happens in embryological development. You have this coordinated signaling among neighboring cells that helps guide them to become all the same cell type, ensuring everything develops as it should. And then finally, let's move on to gap junctions. Direct signaling across gap junctions. Okay, so picture this. You have neighboring cells that have little connections between them. This is called a gap junction in an animal cell, but it's called a plasmodesmata in plants. Anyway, these connections, they're like little tunnels that allow small signaling molecules or ligands to pass between the cells. It's a bit like sharing secrets through like a little passage, right? The special thing about this type of signaling is that it lets cells communicate their current state to their nearby target cells. It's like a neighbor sharing important information. And then this helps a group of cells coordinate their response to a signal, even if only one of them received it initially. And in plants, the plasmodesmata, they're everywhere, turning the whole plant into this giant communication network. So let's not forget about the little signaling molecules, right? The little messages that are being passed around. These are the key players in all of this communication happening between cells. So signaling cells produce the molecules which then bind to receptors on target cells, and these are like chemical messengers that coordinate responses. We call these molecules ligands, and they come in all shapes and sizes, ranging from small proteins to ions, little tiny ions like calcium. Now we have small hydrophobic ligands. These sneaky little molecules can easily slip through the plasma membrane and interact with receptors inside the cell. A famous example of this is a steroid hormone like estrogen or testosterone. They're like special agents that can access classified information inside the cell. On the other hand, we have water-soluble ligands. These are a bit more particular. They're polar, and they can't easily cross the plasma membrane. Instead, they usually bind to receptors on the outer part of the cell's surface. It's kind of like knocking on the cell's front door. The water-soluble ligands come in all sorts of forms, from small molecules to peptides and proteins. They're really quite diverse. Figure 9.4 is showing the difference between internal and cell surface receptors. Internal receptors are found within the cell cytoplasm, and they bind to ligand molecules that can cross the plasma membrane. These receptor-ligand complexes then move to the nucleus and directly interact with the cell's DNA. On the other hand, the cell surface receptors transmit signals from the outside of the cell to the cytoplasm, and there are different types of cell surface receptors, including ion-linked receptors, G-protein-linked receptors, and enzyme-linked receptors, and we'll talk about those next. Okay, so cell surface receptors, we'll talk about those first. They have three main parts. They have the extracellular domain, the transmembrane domain, and the intracellular domain. So let's break those down. The extracellular domain is like the antenna of the receptor. It's on the outside of the cell, and its job is to recognize specific ligands. So ligands like neurotransmitters, hormones, ions. And when a ligand binds to the extracellular domain, it's like a key fitting into a lock. Then we have the transmembrane domain. This part is like a bridge that spans the cell membrane. It can have different shapes and sizes depending on the receptor type. In some cases, the transmembrane domain forms a little pore in the membrane, allowing ions to pass through. It's like opening a gateway for ions to travel in and out. In other cases, the transmembrane domain undergoes changes when it binds to a ligand, and these changes affect what's happening inside the cell. It's like a shapeshifter that triggers different reactions. And then finally we have the intracellular domain. This domain's on the inside of the cell, like the control center. It's responsible for relaying the signal from the receptor to the cell's interior. And there's two ways that it does this. First, through protein-protein interactions, where it interacts with other proteins to pass the signal along. It's like a game of tag, where the signal gets passed from one player to another until it reaches its final destination. Second, in some cases, the intercellular domain itself has enzymatic activity. Enzymes are little workers in the cell, and they can carry out specific tasks. When the intracellular domain has enzymatic activity, it's like having a worker right there in the receptor ready to do its job when the signal arrives. Overall, these three different parts of the receptor work together to receive signals from the outside, transmit them across the cell membrane, and then deliver them to the cell's interior. It's like a complex communication system that ensures the cell can respond appropriately to the signal it receives. Now, before we get too far into the details here, I do want to show a video that gives a nice overall summary of cell signaling, and then we'll get further into the weeds with all of the extra details.
Let's talk about the general sequence of cell signaling. First, reception. Typically, a signal molecule binds a receptor. Second, transduction. The receptor gets activated by this binding. This often means the receptor will change its shape. It could even involve a whole series of molecules changing their conformation in something called a signal transduction pathway. This can amplify the original signal. Third, response. There's some kind of response that is going to happen. A portion of DNA that is found in the nucleus getting transcribed, for example, that's a type of response. Now, cell signaling can involve intracellular signaling, which occurs within the cell itself, and intercellular signaling, where a cell communicates with another cell. In many cases, signaling involves both, signaling between cells and then also signaling within cells. Some cells are close and have direct contact. In the case of these two animal cells, they are gap junction close. Gap junctions in these animal cells, or plasmodesmata in plant cells, are connections between two close together cells that allow ions or other small molecules to pass. And by doing so, they don't have to pass across the cell membrane. Paracrine signaling allows a cell to target another cell by a signal molecule that may diffuse between them. These cells are still close but need not be connected. The ligands in paracrine signaling tend to be rapidly absorbed and rapidly degraded. The ligands are typically not traveling far, as this is local signaling. Synaptic signaling, which specifically involves neurotransmitters in a synapse, is another example of this local signaling. Endocrine signaling can allow a cell to communicate with a target cell from far away. Signals may be carried in the bloodstream. Hormones released by certain types of endocrine cells are a great example. In autocrine signaling, a cell could secrete a certain type of molecule, which then binds to its own receptor and causes a response. A cell releasing its own growth factor could be an example of this. Receptors are frequently part of the cell's membrane surface. If a receptor is sitting outside of the cell membrane surface, then the ligand doesn't have to come in. In fact, there could be properties of the ligand that may not allow it to pass through the membrane. The ligand could be hydrophilic, which would make it hard to pass through. Let's mention an example of a cell surface receptor type, a ligand-gated ion channel. The ligand-gated ion channel is controlled. In this example, the channel is closed. But here comes a signal molecule, a ligand. It binds the receptor, which is the channel protein in this case. Now, the channel protein responds by opening. The ions, not the ligand, go through. Once ions get through, the concentration of ions increases in the cell. And you may wonder, okay, so now there are ions in the cell. Why does that matter? Well, the increasing ion concentration can trigger a cellular response. After reaching a certain concentration and stimulating a cell response, the ligand can leave its binding site from the receptor, and the channel can close. Ligand-gated ion channels can be used by neurons. A neurotransmitter may be the ligand for the channel to open. This could happen at a synapse.
Ion channel linked receptors are a type of cell surface receptor that binds to a ligand and an open channel in the cell membrane. This channel acts like a gateway, allowing specific ions to pass through, and then in order to create this channel, the receptor has a region that spans extensively across the membrane. This region has hydrophobic amino acids that interact with the fatty acid tails in the middle of the membrane. On the other hand, the amino acids that line the inside of the channel are hydrophilic, which means they attract water and ions. Now, when a ligand binds to the receptor's extracellular region, it causes a change in the protein structure. This change, called a conformational change, leads to the opening of the channel, and as a result, the ions like sodium, calcium, magnesium, and hydrogen can flow through the channel. It's like the ligand acts as a key that unlocks the channel, allowing ions to pass through the membrane. G-protein linked receptors bind a ligand and activate a membrane protein called a G-protein. This activated G-protein interacts with either an ion channel or an enzyme in the membrane. G-protein linked receptors have 7 transmembrane domains, each with its own extracellular domain and G-protein binding site. Before ligand binding, the inactive G-protein can bind to the receptor. Once the G-protein binds, it activates and releases GDP, picking up GTP. The G-protein splits into alpha and beta subunits, which can activate other proteins. Eventually, the GTP on the alpha subunit is hydrolyzed to GDP, deactivating the beta subunit, and the subunits reassemble into an inactive G-protein, and the cycle repeats. I did want to show you figure 9.9. This is showing the similar structures of steroid hormones. So these are small hydrophobic ligands, and they have the ability to diffuse directly through the plasma membrane. They have a distinctive structure of fused rings with different functional groups that play important roles as female sex hormones, male sex hormones, and even a precursor for steroid hormones like cholesterol. To travel in the bloodstream, the hydrophobic ligands bind to carrier proteins, and this ensures that they will be soluble and they can transport safely. So in that way, ligands are messengers in the body affecting various physiological processes. Okay, so section 9.1 was discussing the individual molecules and receptors involved in cell signaling. Now let's talk about propagation of the signal and the steps involved in that. So we know that ligand binding, when a ligand binds to a receptor, it initiates signal transduction through intricate networks known as signaling pathways or cascades. So these pathways transmit signals from the cell membrane to the nucleus, regulating gene expression, protein synthesis, and other essential cellular processes. The complexity of signaling pathways arises from the interplay between proteins. Phosphorylation, catalyzed by kinases, adding phosphate groups to proteins, activating or inactivating them. You have nucleotides and other small molecules that also undergo phosphorylation. And then you have second messengers like calcium ions, C-AMP, cyclic AMP, diacetylglycerol, DAG, and inositol triphosphate, IP3, that they transmit signals within the cells and play vital roles in cellular communication. Now, there's a lot of detail in this section and the next section in the book. I'm going to go over the big main important things that you need to know for this course, and don't necessarily need everything that's in the book. So let's talk about phosphorylation first. This is a common chemical modification in signaling pathways where a phosphate group is added to proteins and nucleotides, and it activates enzymes and affects their ability to replace hydroxyl groups on amino acids like serine and tyrosine. This process is catalyzed by enzymes called kinases, and the kinases have different types, and they're named after their specific substrates. So phosphorylation acts as a molecular switch, either activating or inactivating enzymes. And then dephosphorylation reverses the effects by removing the phosphate groups. It's a dynamic process that plays a vital role in shaping the signal. So as we discussed a little bit ago, cellular signaling involves key messengers like calcium, cyclic AMP, and phospholipids. Calcium ions are regulated by pumps in the plasma membrane, and that allows them to be stored or accessed as needed. Ligand-gated calcium channels facilitate the flow of calcium into the cytoplasm, triggering cell-specific responses. Cyclic AMP is synthesized from ATP and activates an enzyme called A kinase, which phosphorylates target proteins. Inositol phospholipids found in the plasma membrane are converted into second messengers. Phospholipase C cleaves them to generate DAG and IP3. DAG activates protein kinase C, PKC, while IP3 releases calcium from the endoplasmic reticulum. This is all very complicated. Just know what the second messengers are and what they primarily do. Again, there's a ton of information about signaling pathway responses in your book, but for this course, I only need you to know the stuff that's right here on this slide, okay? So, cellular signaling pathways respond to external stimuli, influencing things like protein synthesis, metabolism, cell growth, and cell death. Mutations in signaling pathway genes can lead to uncontrolled cell growth, which eventually can lead to cancer. Apoptosis ensures the removal of damaged cells while preventing the release of harmful molecules. And then signal termination is vital for the appropriate responses achieved through degradation and dephosphorylation. So here's an example of apoptosis. It's critical for normal embryology development. So in this figure here, you can see this is a foot and it has a lot of webbing still in between the foot. So this is just an example of programmed cell death where this webbing will be eliminated while the embryo develops. In bacterial signaling, referred to as quorum sensing, bacteria secrete autoinducers, and these are the bacteria's version of the signaling molecules, the second messengers and stuff. So the autoinducers are small hydrophobic molecules, or they might be peptide-based signals. The hydrophobic autoinducers like AHL bind to transcription factors, directly influencing gene expression, while peptide-based molecules bind to kinases, which trigger the signaling cascades in bacteria. Quorum sensing is important for bacterial survival, nutrient monitoring, and avoiding hazards. Biofilm-forming bacteria exchange signals within their colonies, and they coordinate toxin release, and biofilms can pose very serious risks, including infections. Often you will see biofilms forming on medical implants, right? So they can cause big problems for us, but they're very useful in bacterial survival and communication. So figure 918a, this is an example of a biofilm formed by Staphylococcus aureus, and this was found on a hospital patient's catheter. Let's move on to Chapter 10, Cell Division. Cell division is a key process for the growth of organisms. Genes carry the instructions for building proteins and the makeup of an organism's genome. The cell's DNA, which is what the genome is, consists of double-stranded DNA molecule in eukaryotes. In prokaryotes, just like in bacteria, the genome is a single circular DNA molecule called a nucleoid. Some prokaryotes even have additional DNA loops called plasmids, which can be shared among bacteria, and that enables the transfer of beneficial genes like antibiotic resistance genes. Figure 10.2 helps to illustrate the difference between eukaryotic and prokaryotic DNA. Prokaryotes, on the side, they have a simple setup with a singular circular chromosome, while eukaryotes have multiple linear chromosomes that are all wrapped around proteins called histones. In humans, somatic cells have 46 chromosomes, including 22 pairs of autosomes and 1 pair of sex chromosomes. The complete set of chromosomes in these cells is called the diploid state. On the other hand, human gametes like sperm and eggs have 23 chromosomes, forming a complete set with either an X or a Y chromosome. This is known as the haploid state. Genes, or segments of DNA, contain instructions for specific functional molecules like proteins or RNA. The genes determine an organism's traits, and they're inherited from each parent. Duplicated chromosomes consist of two sister chromatids, and during certain stages of the cell cycle, mechanisms come in to compact the chromosomes, and we're going to talk about those. Various proteins play a role in organizing and condensing the chromosomal DNA into a tightly packed structure that's crucial for proper chromosomal segregation during mitosis. When gametes come together during fertilization, they combine their chromosomes to create a new cell with pairs of matching chromosomes. These pairs, called homologous chromosomes, have genes in the same places, determining specific traits and characteristics. So whether it's the color of our hair or the shape of our nose, genes play a big role in making us who we are. Now, like I said, each pair of chromosomes comes from a different parent, which means that the genes they carry aren't identical, even though they determine the same traits. The unique combination of genes inherited from both parents gives rise to the variation we see within a species. Even a slight difference in the gene sequence can result in alternative traits, like blood type, for example. There's 3 possible gene sequences on the human chromosome that determine blood type: sequence A, sequence B, and sequence O. Since we have 2 copies of the chromosome responsible for blood type, our blood type is determined by the 2 alleles or gene variants that we inherit. We can have 2 copies of the same gene sequence, like AA, BB, OO, or from different sequences like AB, AO, BO. While traits like blood type, eye color, handedness may seem minor, they contribute to the natural variation within a species, and these traits may be linked to the expression of other unknown traits. If we compare the entire DNA sequence of any pair of human chromosomes, the difference is less than 1%. The exception to the rule of the identical chromosomes are, of course, the sex chromosomes, the X and the Y, which may have different sets of genes, except for a small amount of similarly needed or gamete production. If we were to lay out the DNA from all 46 human chromosomes end to end, it would measure around 2 meters in length. However, its diameter is only 2 nanometers. Considering the small size of a human cell, around 10 micrometers, DNA must be tightly packaged to fit inside the nucleus while remaining accessible for gene expression. So that's why DNA undergoes compaction during certain stages of the cell cycle. First, short sections of DNA wrap around a core of 8 histone proteins, forming bread-like structures called nucleosomes. This DNA histone complex is known as chromatin. The nucleosomes, along with the linker DNA between them, coil further to create a 30 nanometer chromatin fiber, condensing the chromosome even more. And then finally, the fibrous proteins are involved in the 3rd level of compaction, and they ensure that each chromosome occupies a specific region in the nucleus without overlapping with the others. This way, the packaging of chromatin is efficiently managed within the cell. I'm not going to go into any more detail about chromosome compaction. I do want you to review figure 10.4, which illustrates the steps I just described. I'm going to move on to the cell cycle. The cell cycle is a series of events that leads to cell division. It consists of interphase, mitosis, and cytokinesis. During interphase, the cell grows and replicates its DNA. Interphase is divided into 3 stages: G1, S, and G2. Mitosis is the process of dividing and replicating the DNA into 2 daughter cells. Cytokinesis follows mitosis, and it separates the cytoplasmic components of the daughter cells. So let's go into this a little bit further. This is figure 10.5. It's showing the eukaryotic cell cycle in its entirety. Make note that the cell spends most of its time in interphase. And this is where it's growing. The cells are growing and they're synthesizing DNA. And it's a very small portion of the cell cycle that involves mitosis and cytokinesis. And it's this part that we're going to talk about next. During interphase, the cell prepares for division. It grows, it duplicates its DNA, and then in the G1 phase, we're not dividing yet, we are just accumulating DNA, and we're building up our energy. In the S phase, the DNA replication actually is resulting in the formation of identical pairs of DNA molecules, the sister chromatids. The centrosome is duplicated also in S phase, and that is going to help play a role in organizing cell division. Then we have the G2 phase. The G2 phase, this is where the cell replenishes all of its energy stores, and it also makes proteins needed for chromosome manipulation and movement. Organelles are duplicated in the G2 phase, and the cytoskeleton is dismantled to support the upcoming mitotic phase. The mitotic phase involves the alignment, separation, and movement of duplicated chromosomes into two daughter cells. It begins with karyokinesis, or nuclear division, which consists of several phases. The phases of mitosis that you probably have heard before, these are actually the phases of nuclear division. And they are prophase, prometaphase, metaphase, anaphase, and telophase. So let's look at those. G2 of interphase, again, that's where the cell is replenishing its energy, it's making proteins, all that kind of stuff. It's duplicating the organelles, right? Then in prophase, the nuclear envelope dissociates into vesicles, organelles are fragmented and dispersed, the nucleolus disappears, centrosomes move to the opposite poles, and microtubules form from the mitotic spindle. The sister chromatids are visible. So these are the chromatids, these are the spindles, the centromeres are in the center of the chromosomes, okay? Or the chromatids. Then in prometaphase, this is the first change phase. This is where sister chromatids bind to the spindle, and then chromosomes are oriented and attached to the microtubules from opposing poles. The polar microtubules overlap and contribute to a cell elongation, and then the astral microtubules near the poles aid in spindle orientation and regulation. So it's building, prometaphase is where all of this structure is coming into play, and it's helping to line those sister chromatids up, okay? Then you have metaphase proper. This is where the chromosomes are aligned at the metaphase plate or the equatorial plane. In other words, they're lined up right in the middle. Sister chromatids remain tightly attached. And then in anaphase, the cohesion proteins degrade and it separates the sister chromatids at the centromeres. And then the chromosomes are pulled towards the centrosomes. So in metaphase, everything's lining up in the middle. Anaphase, everything's starting to split apart. And then telophase is where the chromosomes reach the opposite poles and they decondense. The mitotic spindles depolymerize, and then the nuclear envelopes form around the chromosomes. The very final stage, of course, after this is cytokinesis, and that's where these two cytoplasmic membranes become separated. Here I have another short video that helps better illustrate mitosis.
Most eukaryotic cells follow a process of growth and division called the cell cycle. These events include a growth stage, mitosis or nuclear division, and cytokinesis or division of the cytoplasm. Throughout interphase, the cell is engaged in growth and metabolic activities. Interphase can be further broken down into three discrete phases called G1, S, and G2. During the G1, or first growth phase, normal cell functions occur as well as cell growth. In the S phase, DNA replicates, producing 2 copies of each chromosome. During the G2 phase, the cell continues to prepare for mitosis and cell division. Mitosis, or the M phase, has 4 stages: prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes become visible and condense, becoming shorter and thicker. Each identical copy of a single chromosome is called a sister chromatid. The nuclear envelope breaks down and spindle fibers form as microtubules grow out of the centrioles that move to opposite poles of the cell. During metaphase, the double-stranded chromosomes line up along the equator of the cell. The spindle is now fully formed and the microtubules attach to each sister chromatid. Anaphase begins when the sister chromatids of each chromosome begin to separate. The centromere that holds sister chromatids together divides, and the chromosomes move away from each other along its spindle fiber. In telophase, the two groups of chromosomes reach the opposite ends of the cell. As a new nuclear envelope starts to form around each group, the chromosomes uncoil and the spindle disappears. The division of the cytoplasm and organelles is called cytokinesis, or the C phase. The result of mitosis and cytokinesis is the formation of two genetically identical cells.
Now finally, let's get into control of this cycle. So if you remember this figure here, right, this is the full cell cycle. Controlling this, right, there's a lot that can go wrong during cell division. And that is why each step of the cell cycle is monitored by checkpoints. There's 3 major checkpoints. There's one in the G1, one in the G2, and one in the M metaphase. Okay, so let's go through those. The G1 checkpoint determines favorable conditions for cell division and irreversibly commits the cell. Factors like growth factors, size, reserves, DNA damage, these things are all assessed during this checkpoint. If the requirements are not met, the cell halts the cycle or enters G0 until conditions improve. The G2 checkpoint prevents mitotic phase entry if conditions aren't met. Cell size, reserves, and accurate DNA replication are all assessed at this checkpoint, and it's to ensure that the DNA integrity is intact, and it allows for repairs if needed. The M checkpoint confirms proper attachment of sister chromatids to the spindle fibers, and this is crucial for irreversibly separating chromatids during anaphase. The cycle proceeds when each pair is securely connected to spindle fibers from the opposite poles. Not all cells follow the typical cell cycle pattern of immediate entry into interphase followed by mitosis and cytokinesis. Cells in the G0 phase are not actively preparing to divide, they're in an inactive stage. So this can occur when cells exit the cell cycle either temporarily due to environmental conditions or permanently in cells like mature cardiac muscle cells or nerve cells. Cells in the G0 phase remain in this phase until conditions improve or until an external signal triggers the start of the G1 phase. Sometimes the cell cycle goes wrong, resulting in uncontrolled cell growth and formation of things like tumors, or it can even cause cancer. Cancer occurs when cell division becomes unchecked due to the breakdown in the regulation of the cell cycle. Cells, for some reason, will bypass a checkpoint and divide excessively, leading to the formation of tumors and masses. The loss of control starts with changes in the DNA sequence of genes responsible for regulating the cell cycle. These changes result in dysfunctional proteins, and when the monitoring system is disrupted, errors can be passed on to daughter cells, and with each new cell division, more damage accumulates in more daughter cells, and eventually all the checkpoints become dysfunctional, and rapidly dividing cells overpower the normal cells, forming tumors or even causing leukemia. Tumors can be benign or malignant, and benign tumors, they stay localized and they don't invade surrounding tissues, but malignant tumors invade nearby tissues and can spread to other parts of the body, and this is what leads to cancer. I did want to show you a short video.
More than 60,000 billion cells make up the human body. They are the units that form the tissues which make up our organs, like the liver, heart, and lungs. When the body needs it, our cells split into two and replace those cells that are defective or have come to the end of their lifespan. This makes it possible for our tissues to preserve their shape and their respective functions with the passage of time. Every cell is therefore programmed to multiply and die. This ordered but complex program is controlled by the center of the cell, the nucleus, which contains chromosomes containing many genes made up of DNA. Sometimes, some of these genes undergo a change. The nucleus then sends out abnormal orders and the cell goes wrong. It multiplies uncontrollably and takes on a life of its own. Each new cell produced contains the same defect. The cells proliferate chaotically and form a tumor. This process may be short, but is often long. 10 to 30 years may separate the birth of a first abnormal cell from the appearance of a tumor of about 1 cubic centimeter. The tumor forms many blood vessels in order to survive. These will supply the tumor with oxygen and nutriments, allowing it to live and grow. This is what we call the phenomenon of angiogenesis. But why does a cell become cancerous? There are many factors, and they can exist to different degrees. Factors have been identified, such as hereditary genetic anomalies, exposure to some viruses, HIV, hepatitis B, C, and D, papillomavirus, Epstein-Barr virus, exposure to toxic agents, chemical products, radiation, sun, unhealthy behaviors, such as the consumption of alcohol and tobacco, or a diet too rich in fat and low in fruit and vegetables.
Finally, let's go over prokaryotic cell division. We'll briefly discuss cell division in prokaryotics. It involves replicating, again, the genomic DNA and distributing copies to daughter cells, just like in eukaryotes. Prokaryotes and eukaryote cell division is very different because prokaryotes, again, they only have the single DNA, while eukaryotes have organelles and they have multiple chromosomes and things. So prokaryotic cells divide through binary fission. During binary fission, the DNA replicates, the cell elongates, and eventually the cell membrane pinches off and divides the cell into two daughter cells. Bacteria, with their single DNA chromosome, they don't have a nucleus, so they don't have to undergo mitosis like eukaryotic cells. Bacterial cytokinesis is guided by a protein ring called FTSZ, but the ingrowth of the membrane in the cell wall forms a septum that constructs separate cell walls for the daughter cells. Let me show you a very quick video on how this looks.
The process of prokaryotic cell division is called binary fission, which is a simpler process than found in eukaryotes. Recall that prokaryotes differ from eukaryotes due to the lack of a nucleus or cell organelles. Typically, their chromosomes consist of a single loop or circle of DNA. The circular DNA molecule replicates. It then attaches each copy to a different part of the cell membrane. The cell grows to twice its size and the membrane pinches in the middle. The cell can now undergo binary fission. Binary fission is the prokaryote form of asexual reproduction. After the cytoplasm divides, each daughter cell synthesizes a new cell wall, creating two identical cells. Binary fission results in two cells that each contain one copy of the original circular DNA. Prokaryotes are able to multiply rapidly because of their small size. Their DNA takes a short time to replicate, and with a high surface area to volume ratio, they can absorb nutrients quickly. Also, the relative simplicity of the cell's internal organization allows rapid duplication. A bacterium can divide every 20 minutes in optimum conditions. Unchecked, bacterial population growth is exponential, allowing rapid colonization of a favorable environment.
Okay, we covered a lot of topics today, including cell signaling, cell division, cancer, prokaryotic cell division. I want to thank you guys as usual for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.